Okay, let's get started. So we're still on two-dimensional data, but so far what we've been dealing with primarily is sort of quantitative data that's real valued. So that we've been looking at positional data effectively, so we can draw scatter plots. I'd like to sort of turn attention now for a while to categorical data and how we might visualize that. And just to remind you about categorical data, it comes in a few varieties. One is just plain nominal. So these are the scales, nominal or ordinal. And nominal, I mean that the categories just name something. So there's no ordering between the category values. For example, Canada, United States, and Mexico, just three names of categories of things, maybe companies, where they're located, etc. Sort of timely talking about NAFTA, I guess, that we've got these three countries, but they're just names. Or it might be color, red, orange, yellow, just to identify that's the value of a variate. Or maybe the party for which you voted, so that in Canada that might be Conservative or Green or Liberal or NDP or some other party. And this will be contrasted with categories where the values can be ordered. One seems to be smaller than the other. So for example, opinion, you're often asked when you're doing course evaluation, for example, you're asked to give a score, where the score, the value itself doesn't mean anything, but it does provide a ranking. So an opinion might be on a rank from disagree, somewhat disagree, no opinion being the neutral in the middle, the zero, somewhat agree, and they're clearly ordered. Similarly, color itself might be in order if it was actually looking at the spectrum. He was looking at the rainbow, that's when it might be in order. And other things that you've seen on surveys. So if you only have categorical data, and that's what we're going to look at for the moment, you only have categorical data, principally what we're interested in is counting how many of each value occurs in the data set. Okay, so we're just about counting, and combinations of the values are also interesting. How many in each combination? Variates themselves also come with different kinds of constraints on them, largely brought about by how the data were gathered, sometimes by the nature of the data itself. So the ones we want to distinguish here are where there are no combinations are possible. So imagine I've got two variates. One is data visual, what course are you registered in, if you're in this course, right? So I may know how many of you are registered in Stat 442, how many are registered in Stat 842, how many are registered in Comp Math 762. I might know those counts. And then I might ask, well, say, what's your citizenship? And we could do a show of hands by, is your citizenship of this country or that country or another country? And we would have counts for those as well. But I don't have the combination. I don't know which citizens were in Stat 442, etc. So I don't have both of those together. So I just have the marginal information on each of those. So data sometimes comes like that. You've just got the marginal data itself. Another possibility is that the data values are actually hierarchical. So we may be looking at countries, for example, and think about, what country were you born in, right? So there's going to be a list of countries that are available. What state, province, region, whatever name is for that country, were you born in inside that country? What city, village, etc. The values of those variates, if a variate is the state, is the country, it can only take so many different values. The variate is then what state, province, it can only take so many values, and the values it can take are constrained by which country you're in. So there's this natural hierarchy. Okay? So sometimes the data is completely nested in that way. There's hierarchical arrangements of the variate values. Then there's everything's open. We've got non-nested data without any restriction, and we may have some parts of the data, not other parts. So for example, we may have lots of different species of plant or animal something, and we have the all of these genes, and each gene we know whether it belongs to, it's shared by which species, right? So for every gene we know that it's in this species or that species, etc. And what we're interested in, what is the, what genes are in common between species, what genes are different between species, etc. So this non-nested without restriction often comes up in modern data, thinking about just what is there in the data set, what is the distribution of these genes. The genes are the counts, effectively. And finally, what we see a lot of, particularly in analysis of data and often design experiments, of course, are completely crossed variates. So I have all of the values on, say, 2 variates. If we look at the Titanic data, I have all of the passengers, and for every passenger I know what class they were on the ship and what their sex was. So I know both the class and the sex for every passenger. And those 2 variates, class and sex, then I think of those as being crossed. So they're completely crossed. All values of one can occur with all values of the other, and I can have counts on every cell in that cross. Okay? So those are the different structures that we're just going to look at over today in the next lecture. Any questions on the structure of categorical data? It's pretty rich, even though it's only very few values that the variate can take. Okay, let's start with just marginal data. So all you've got is marginal information. And if all you've got is marginal information, so 2 plots that you've used since, again, grade school would be bar plots and pie charts. Right, so here I'm going to look at voting in the last Canadian federal election. And so I have which party you voted for: Bloc Québécois, Conservative Party, the Green Party, the Liberal Party, the New Democratic Party, or some other party that's not there, too small to be mentioned. You know, I guess the Marijuana Party, the Communist Party of Canada. Lots of other, I don't know if we have a Libertarian Party, maybe we do, but I'm sure there are lots of other little tiny parties kicking around. They're all lumped into other. And then I have the percentage of votes that each of those parties received in the 2015 election, and then the number of seats in the Canadian Parliament that those votes turned into. Okay, so in R, I'm just going to use a bar plot or a pie chart, the pie command. So let's have a look at the results here. So if I look at the bar plots, so the 2 variates I have are political party voted for and political party that the seat went to. Okay? So on the left you see the percentage of the vote that each of these parties received, going from 0 to 30. This red one is the tallest, that's the Liberal. They received the most votes at just under 40% of the votes. So the question you might ask is, what proportion of the total votes did the Conservatives get? Well, you can identify that fairly quickly by just reading across the axis. So we have positions along a common aligned scale. So it's very easy to read these values off. Okay? I can ask you another question, though. If I look at this, what proportion of the votes were gained by the Conservatives and the Bloc Québécois and the Green Party combined? That's a little more work, right? You're going to have to read these off, maybe add them up, or imagine stacking these 3 things on top of each other to see whether that's, and then read off the total at the end. On the right-hand side, we can look at how many seats do you get. And it's the same sort of thing that the largest number of seats go to the red bar, the Liberal gets the largest number of seats, Conservative second amount, hardly in the Green, and none for the others. No seats in Parliament. We can identify number of seats easily by reading off a common aligned scale, right? And we can still have the problem of thinking about, well, do the Conservatives, do the Liberals actually have a majority in the Parliament? And what's that mean? Well, the majority means is that red bar taller than the height of the sum of all bars. Okay, so the height of stack the Bloc Québécois on top of the Conservative, the Green on top of the Bloc Québécois, New Democrats on top of the Green Party, and whatever's another on top of the end. Is that bar taller than this? It's a little difficult to tell, right? So think about, when you're looking at that, what are you going to think about the comparisons? The other thing you might ask is comparing these two. It's clear we don't have proportional representation in that the arrangement of the bars on the left here are different heights than the bars you see on the right. It's scaled here, so the red bar is still the tallest in both, but the blue bar has shrunk, the orange bar has shrunk, every other bar has shrunk. So they aren't getting the same number of seats in Parliament as the proportional of the vote. Okay? All right, so that's reading into a bar plot. There's a lot there, and think about the graphical perception laws that you know about to assess the quality of this particular graphic. Let's look now at the pie chart. So the pie charts on the left are the percentage of vote, and on the right are the number of seats. On the left, the percentage of votes, you can see that certainly the Liberals did not get anything close to half of the vote. They don't get anything close to 50% of the vote. In fact, the sum of all these now are easy to compare. The sum of all these is much larger than this Liberal vote. So roughly 60% of people voted against the Liberals. Right? 60% of the voters chose not to vote Liberal, voted for some other party. And you contrast that to the right-hand side, where it's again pretty obvious that they have greater than 50% of the seats in Parliament and hence form a majority government. Okay? It's an artifact of our first past the post voting system. So Green Party over here, which has a visible chunk of the vote on percentage of the vote, has effectively nothing in Parliament. They may have one seat. The Bloc Québécois, which is a fairly big chunk, there, of course, nobody's voting for Bloc Québécois outside of Quebec, right? So these votes are concentrated in ridings within inside Quebec, so they're going to have a much smaller choice. It's not that anybody wouldn't vote for them outside of Quebec. There may be people that would, but they don't run candidates outside of Quebec. And similarly, these pieces of the pie, you can see, are all smaller than those pieces, just like you could with the bar charts. So when you think about, well, what is it easy to tell with this pie chart? Is there anything that's easier to tell than with the bar chart? I think that's certainly easy to tell. We can tell that that's a little more than 50%, that that's not a straight diameter going across that plot, right? I think most of us can pick up pretty quickly that that's bent. And so there's more than 50%. We can tell the difference between a half or not fairly easily. We can also tell sum things up, provided they're contiguous pieces. We can see which pieces are bigger, which sum of pieces are bigger compared to trying to do it with that graphic. Okay? And if you recall the graphical perception experiments that were done by Cleveland and others, when they looked at angles, right, trying to recover the measurement by encoding it with an angle, that performed very poorly compared to length or position along a common aligned scale. So just on that evidence alone, often it's the case that people will dismiss pie charts as being silly things because there's always a superior graphic. However, recall that those experiments are assessing one thing: how accurately can I return that value, that encoded value? And there may be some value here in looking at sums or comparisons of different pieces together. And certainly it's easy to tell whether it's a half or not. So if you're interested in the American election, 2016, we have the percentage of the vote on the left and the number of electoral college seats. This on the right is what determines the presidency. So they also have in a system where the popular vote, the percentage of popular vote, does not determine the presidency. Principally, they have a middleman called the state. What you're voting for is how should your state vote? And again, that's most of the time first past the post. All the electoral college votes for that state go to the winner, no matter how little they may have won by. Okay. On the left, did Clinton get half of the popular vote? Show of hands. How many people think she got half the popular vote? How many people think she did not? Yeah, she didn't get half the popular vote. You can't see that that's bent. That's not a perfect diameter there. This, from here to here, is not a straight line. She does not have half of the popular vote. She's got, I don't know, 48, 49% of the popular vote, but not 50%. And Trump has 40 some odd percent of the popular vote. And the others, Libertarian, Green Party, who else is there? Some other parties have a smaller fraction, about 5%. So again, that turns into, it's clear that Trump has much more than half of the electoral college votes to establish the presidency. So there are perceptual experiments that look at comparing these things that go back to Lewandowski and Spence, looks like nearly 30 years ago. Think about the perception of statistical graphs. And so they're comparing a pie chart versus a bar chart versus a table of the same numbers, and then asking these questions. What is the relative size of some category values, A, B, C, etc., to others? So A versus B, and which is larger, A versus B plus C, A plus B versus C plus D. So you can imagine if you think about trying to do that yourself for any one of these things, which of these are easy to answer and which are the easier graphics to use with. Okay, so they do that kind of experiment. The results are, in terms of accuracy, again, following along like the Cleveland et al. Thing, but the accuracy, as this thing gets more complicated, A plus B versus C plus D, where you're adding chunks together, the pie chart actually outperforms the bar chart and the table, which is kind of surprising. But maybe not so much when you think about it. Particularly their comparisons are often contiguous, right? They also made these comparison of these different representations. Just two numbers here, a table, this length or position along a common scale. The question, yes?
Yes. So for the table, wouldn't that be the most accurate? Because you just add the numbers and do a division.
Yeah, it turns out not. So the question is, since we're recording this question, shouldn't the table be more accurate since you're just adding numbers? You might think so. So things that you might measure are accuracy while you're doing this, and also how long it takes you to do. So latency. But this is entirely accuracy here, percent accurate. So on the people, it's a human experiment. So on the people that you studied, right, your sample, you conducted this, the pie chart outperformed the table when it got complicated. So I guess I'm adding two numbers comparing to adding two numbers. I'm adding two numbers twice and looking at the ratio, let's say. So it seemed to be that they got a better accuracy with these folks than when they just looked at the pie, relative size of the pie pieces.
So I assume when they do the experiment, they don't allow you to
No, no. They don't allow you to bring a calculator. They don't allow you to bring a protractor to measure the angle of the pi. You're just there you're looking at it and you're going to tell me what the answer is. Okay? So same sort of experiment here. We're looking at table 2 numbers. This the height of these bars, size of this pi, this now volume, which is really still essentially the height of the bars, but it's encoded to look like a volume. Same thing here with a cylinder. And length, vertical length, horizontal length. Again, they're positioned with this along a common scale. And then this is kind of representative of a 3D pie chart. It's a disk that's tilted away from you a little bit. If you drew a little thickness around there, it may look a bit like a 3D pie chart. So that disk. You're asked to compare the relative sizes of A and B. And what they found was the following. The things that they're recording is the error, the percent error, so smaller is better. Latency, how long does it take you to get that value, so smaller is better. And the exponent, and you remember these are the exponents from the power law, right? So Stephenson's power law, so a value near 1 is what you like. So for these things, for the bar, the pi, and the disk, the exponent is all about 1, which you would expect. The error rate for the bar and the pi are comparable. Pi is a little smaller error than the bar. don't have standard deviations here to have a sense of how variable that is. Disk is much larger error. Latency, all three are comparable, with the bar being the fastest and the pi being the slowest. And these other things just don't quite perform as well. The exponent, as we know, when things look like it's volume, don't behave as well. And let's see, error percentages are a little higher. Table again, now it's consistent with your observation. You've only got 2 numbers, right? What's the relative size of these numbers? That accuracy is the best for the table. The latency, the time it takes you, is a little longer, 8.6 seconds on average. Interesting thing here is that the vertical line has a lower latency in evaluating the result than the horizontal line, but the error is a little higher. So I don't know whether we should read too much into that, looking at things that are vertically differences, how important they might be compared to horizontal differences in whatever our evolutionary path was. But there's not again, we don't have the standard deviations here to say how variable these might be. Okay, so that's the simplest thing where I've got nothing in common. Now let's look at a complicated one where I've got hierarchical data. And the hierarchical data looks like this on the left-hand side. Think about this as a diagram, but as a tree. And so at the top I've got this grouping A, so this may be the country, state, city, street, or whatever inside. So I've got A has 16, so there's 16 counts in the whole thing here. 16, that gets split into 3 pieces, subcategories. 3 here, 10 there. This one is split into 2 categories, 1 and 2. This into 3, 2, 4, and 4. And they get separated down here to this 4th one. It's got 1, 1, 1, 1. And the idea is to get a visualization of this hierarchical structure that conveys the number, the amount that's in each of those categories. Okay? And the proposal is a tree map, and you've probably seen these on the internet a fair bit. Lots of sites like to have tree maps. And the idea is fairly straightforward. So if you look at the tree on the left and the tree map on the right, we're going to be cutting this vertically and horizontally, and with the areas of each of these rectangles being proportional to the count here, or as best we can make it proportional to the count. So the first cut is from A, the whole group, into 3 groups. That's going to happen cutting along along the vertical axis, just cutting these horizontal axes into 3. So that's, this is B, that's C, and the rest of this is D. So there are 10 things here. Then once I have B, I've got 2 groups here. I'm going to cut it along the horizontal, cut it across the vertical axis. E has 1 and F has 2. So it's a third of the way along. Okay. C, there has no children. D now has 3, so it gets that group, and that group. 3 cuts. This leftmost group gets cut into 2. Middle group has no children. The last group gets cut into 4. And hopefully the areas of all of those rectangles are proportional to the counts. Okay? Everybody understand a tree map? Yes?
Does the letters, do the letters only represent like a category or something?
Yeah. Yeah, I mean, this is the most abstract version. I'll show you some real data in a second. So the letters are there, and the number is just the count in that category. So you've got a category and subcategories. Okay? But there's no crossing over. You don't have an E under B and under C. So it's entirely hierarchical. Okay? Any other questions? So if you understand this picture, you understand tree maps. Let's figure out the algorithm of how you'd actually do that, which is where the work is. All right, so let's look at that olive oil that you've been looking at with the assignment. And you know that we've got regions, north, south, and what was the other region? Sardinia, I think. And then you've got areas within those regions. In Italy, these are places where the olive oils come from that we're looking at. So I'm going to count, there's a natural hierarchy of this areas within region. And I'm just going to count how many olive oils there are in each one of these areas and hence within each region. Okay, and construct the data set. So I construct that data set, and that data set's going to look like this. So the counts look like that. I've got 3 regions, South, Sardinia, and North. Within South, I've got North Apulia, Calabria, South Apulia, and Sicily. Sardinia, inland and coastal Sardinia. North Umbria, East and West Liguria. Okay? And that's the number of olive oils in that data set that are inside each of those areas. Okay? That's all this work was on the other slide, just to create that. Okay? So now you see that I've got 2 categorical variates. I've got a region and I've got an area. Region can only take these 3 values. Area can only take this many values. Right? 1, 2, 3, 4, 5, 6, 7, 8, 9 values. If I counted those right. But these values can only appear, North Apulia can only appear a match with the South region. It can't appear matched with Sardinia. It's not in Sardinia. So there's hierarchy there. Okay? So there's a package called Treemap. There are lots of packages, as you might guess, in art that many do the same sort of thing, but here's one that's called Treemap. And so I take these olive oil counts, that data set I've got. Index is going to be region, area, and the size that I want is the count. Right? So I'm looking at, that's the count. That's where I'm getting the size of these areas. So what's happened? This has been divided into, first of all, this whole left-hand side got divided by this vertical line, so Sardinia got peeled off here. Then within Sardinia got into coastal and inland. The north, unfortunately, ended up on the bottom, which is backwards for us if we think about maps. But north is on the bottom. This got sliced across this way. Okay. And then once inside, the north got sliced this and this. And the south got sliced into these 4 pieces. So that's not quite what we had a minute ago. Not quite what I just told you of a way to do this. So we're trying to be a little cleverer, right? To try and, because we have to get these areas to match the actual count. So a little clever of thinking about how can I tile these 3 big areas first, right? So that I get some sort of aesthetic pleasing. So you can imagine if you go look at the Treemap code, there's going to be some objective function there that's trying to be minimized to try and make this pleasing. Okay. Now, even with this, sometimes you end up with these long, skinny things. And what we'd like to have, or what other authors have suggested, is it'd be nice if these were closer to square. That is, if the aspect ratio of all these boxes was as close as to 1 as you could make it. Not going to be possible and be able to tile this for them all to be 1, but you want to get that close. So now you've got a slightly more complex objective function in how to actually find at least a local optimal solution. So, hang on, fun begins. I was going to show you this. Treemap actually lets you do an interactive one. I'd forgotten that. So here's our data, so you can actually play around with this. So there's the first cut. Now Sardinia is on the bottom in this one instead of on the right-hand side, so they flipped this around. In region, if I did area instead of region, so you see that coastal Sardinia is here, inland Sardinia is over there, so we're not respecting the regions anymore. So turn that back into region. This is a Shiny app, which are pretty easy to produce in RStudio. And that's what we have on the default. So having region, then area, and I'm looking at the count. So let's see what happens if I don't fix that. Area. Looks like it's producing pretty much the same anyway. Okay, so an interactive version. So Treemap. But back to the point I was just making, that aesthetically you might like to have those be squares so that they're a little more comparable. Comparing areas of things that are roughly the same aspect ratio is better than comparing areas that are different aspect ratios. So there's another approach which you can look at in this paper from 99. And another R package called Treemapify, and it uses ggplot, and the most recent release is really heavily ggplot. So we're going to get a tree map with a ggplot with the aesthetics being right, area, fill, and a subgroup, right? And then Treemap plot, just take that ggplot and add these various things, various geomes. And we get this result. And as you see, it's trying to make these things so there's no long, skinny Sardinia here. Sardinia is a little, got it closer to an aspect ratio of 1 than it had in the previous one, where it was just slicing off one end of the square. So that's what Treemapify is trying to do, is try to get these aspect ratios close to the same. I mentioned here, there's also, if you look at the Google Viz package in R, you can also find a Treemap functionality. Changing things up here slightly, I can get a color now for every one of the areas, so the areas are themselves more distinguishable. And some of those geomes were all about putting boundaries around each of the large regions and labeling the pieces. This is a nice effect, at least, that these labels seem to be out of each other's way compared to Treemap. Okay, so that's Treemap, and you can imagine thinking about how could I improve on that? How complex might the algorithm be to try to figure out a nice layout? But it is going to be this recursive laying out by layer. Finally, Treemapify also does you the courtesy of, if you don't want to use their software, but you just want to know where do they think the boxes will be, you can get the coordinates just from Treemapify itself, a function. So you can get the coordinates of all of the rectangles. Okay. Any questions about tree maps? 'Cause we could spend a fair bit of time if we actually looked at the algorithms. But I just want you to see that there's lots of opportunity for hierarchical visualizations. And how many people have seen tree maps on the web? Oh, that few. I thought everybody would see. It's often, you'll often see that as a result of searches to see what's going on in a particular area. So it's a nice way of representing hierarchical data. So, non-nested data. And think about a common visualization here is just intersecting sets, Venn diagrams. Again, something you've known for a long, long time. And what's curious is how, with modern science, how the demand for Venn diagrams have been increasing. So these are just looking at articles since just before, I guess that's 98, of two famous publications, Nature and Genetics, online versions, and just how many articles mentioned are using a Venn diagram. And this is increasing substantially over time here, the last decade or two. Right? So Venn diagrams are appearing more and more and more, and largely because of the nature of the data that scientists are encountering. So here's an example. You might be interested in the contents of a database and the contents of any group of things, just what's in there, what's in that particular database, tell me about it. And just a quick summary of it. So here's a genetic database for the number of genes shared by various cereal crops. The cereal crops are these four: rice, maize, sorghum, and Arabidopsis. And in that database, we know how many genes there are and whether those genes are shared by these various crops. For example, there are 265 here. Oh, I can't select that. 265 here that are just sorghum and nothing else. 465 that are just maize. 405 that are shared by both maize and sorghum, but neither rice nor Arabidopsis. All right. Now this is what's in that particular database. It may or may not be complete on the whole list of genes that these crops have. And again, if we're looking at, there's 8,494 of them that are shared by all four of these cereal crops. Okay. The claim is that that's kind of hard to convey the volume, how much of the database. I mean, you've got visually for that over 8,000, you've got roughly the same visual real estate allocated to it as you've got for 34. So could you give a visualization that was a better sort of gestalt, hit you in the face of how much do these various crops intersect in their genetic, the genes they share? So what we'd like to have, and the other claim is, and seems to be true, people certainly do like to have, if they can get away with it, circles for Venn diagrams. Whether that's just a natural thing or that's something that we've been trained to, but certainly, at least for small numbers of groups here, the circles are often used. So there's a package called Venn Euler in R, which attempts to use circles. And what we want is the circle area to be proportional to how many things are in that group. So the area to be proportional to what's in that group. But the hard part is we'd also like every intersection to have its area proportional to the count in that intersection. So you've got circles that you can change the radius of, and you can move their centers around. And they'll intersect a certain amount and not. And every one of those areas of intersection, you'd like that to be close to the actual count in that intersection. Clearly it's constrained. You're not going to be able to do that exactly. But you might get close. Okay? There are several other more recent packages in R, including one developed locally called Venplot that tries to solve this problem, and I think all are an improvement on Venn Euler. But Venn Euler is the thing that started it, so I wanted to highlight it. So, for example, so here's a genetic database of the number of genes shared by 12 animals. And this is a solution by Venn Euler. And you can see that the idea is that every circle is proportional to the number of genes in this database, not all the genes in the animal, number of genes in this database that animal has. So chicken has a large number of genes. So you can see immediately in this database, it's got lots of genes from a chicken, lots of genes from a salmon, lots from a pig, right? Not so many from a rabbit. Some from a frog, not so many from a horse. But it's not saying that there are more genes and the salmon has more genes than a horse does. It's just what's in this database. And it's also not saying that a pig has no genes in common with a rabbit. That's not true either. It's just what happens to be in this database. But all of those areas, the intersections here, so humans and dogs, the intersection of our genetic, of the genes in this database that are shown by, are tried to be matched by that area of intersection. So humans appear here. The only genes that we, in this database that are in common between these four groups are humans, chicken, cow, and dog. And it doesn't look like there's very much that is shared by chicken, cow, and human in the database. But the algorithm is trying very hard to ensure that each area here matches the count in the database. So the idea is that with that view, you get a quick sense of at least what's in this database. Right? And we all know that actually the intersection, if we had all of the genes of all of these creatures, the intersection would be much, much larger. Okay. Here's another example. You can look at, for example, English literature. These are unique words in common. So there's a number of unique words in each one of these things. So you've got the Bible, Ulysses, Macbeth, Silas Marner, and Moby Dick, and Dorian Gray, some famous novels, famous play, and famous book, the Bible. And the idea is, can we get an arrangement so that what they have, the number of words they have in common corresponds relative to the area. So clearly here, Ulysses got a ton of words, unique words that are not appearing in these other things. There are a lot of different words in Ulysses. Now, you imagine statistically how you'd be assessing the fit here. The fit would be such that let's look at every area that appears here visually and compare that to the count. And you can regress one on the other, right? Or you can see how correlated are those two areas. I've got the counts, how many words there are in each one of these intersections. So how many words there are that are both in the Bible and in Ulysses, but not in Moby Dick, right? So that there, I've got an actual count, and there's a visual area here that it can be measured. So when you look at this arrangement actually gets a correlation between those two measurements of nearly 1. So that's a pretty good representation. Okay. Any questions? So just this thing about the nature of the Venn Euler thing, when you're looking at it, what's identifies is if I give A, B, and A and B, these numbers, that's what's in A that is not in B. Okay? This is what's in the intersection. So roughly these are a third, a third, a third. 1, 1, 1. So it's just a peculiarity of the interface. So another example here, again a genetic one. So this is what's published in a scientific paper, 13 genes here, 14 there, 91 there, 101 here. And the genes are by these various labels. And the argument is that instead of publishing that, just publish this, and you see far more immediately what the intersection is and how many genes there are in that particular collection. Okay? So everybody get the point? Yeah? All right, just start, I guess, on cross data. So now what have we done? We've got marginal data, hierarchical data, and then sort of these intersections counts using a Venn diagram, and now cross data. And so what I think about it here, I've got two variates. These are a bunch of students from the University of Delaware, and we have their eye color, brown-eyed, blue-eyed, hazel, green, and their hair color, black, brown, red, blonde. So for every person, we know their eye color and their hair color. So that's what I mean by it's crossed. So I have this whole 2x2 table, not 2x2, I have this whole two-way table, here 4x4 table. Okay? So just think about, we could talk about table layouts of this thing, but we're thinking about other visualizations. And what I'm going to just, I guess, have time just to introduce to you is this something called an eikosogram. And this eikosogram is meant to be a drawing of probability, a drawing of proportions. And so instructions here, this will be placed on the class website, and you can just download it, place it somewhere, and then install the package in R. And you install that package in R either from sort of menu system. It's install packages with a package and archive file that you're picking up, and then just browse to wherever it is and hit install the library. Or if you've got a Macintosh or some sort of console that you can do, you can actually run commands, just change directories to wherever it is, and do R command install the thing. I also believe that what will be on the website will have a .gz. I think I gzipped it too. Questions? So those are instructions. You can follow that quickly, but let's just get an idea of what this is. So cross data. An eikosogram has in mind that one of these variables is a response and the other is explanatory. In that sense, it's kind of like a regression, okay, or a response model more generally. And it's going to divide the unit square, much like a tree map. It's going to divide it into vertical slices and horizontal slices. So what I have here, I've got, for this data set, I know I've got the hair color determination and I've got the eye color determination. And so I choose one of those variates and imagine that I'm going to look at hair color, explain it as a function of the eye color, right? That being the case, I have a response variable, that's the hair, and the conditional variable, the part that's going in this conditional part here, as the explanatory variable. So that'll be I. Okay? And I think I'll just show you what this picture looks like. So it's easier to explain how it's constructed when you look at this. So here's an eikosogram. And on the vertical axis is the y, on the horizontal is the x variable. So this is the response, and this is the conditioning or explanatory variant. So it's I on the bottom here. We've got 4 different eye colors. The width of this first bar is proportional to how many people have brown eyes in that group. This width, how many people have blue eyes, how many people have hazel eyes, how many people have green eyes. And then within the group that has brown eyes, the proportion that have black hair will be that height. The proportion that have brown hair is that height. The proportion that have red hair is that height. And the proportion that have blonde hair is that. And we do that for each of the eye colors. So you can imagine that if I see brown eyes, it looks like the probability of having blonde hair given brown eyes is pretty small. So the height of each one of these rectangles is a conditional probability. It's a probability of brown hair given brown eyes. Over here on the rightmost top right corner, the height of that rectangle is the probability of having blonde hair given green eyes. Okay? And the width of each one of these rectangles is the marginal probability of that x. The marginal probability of having brown eyes is that width. The marginal blue eyes that width. And you imagine the whole thing is a unit square. So that's an eikosogram, and it really plays a role as a response model.
Okay, so that is here is the response, and I is the conditioning variate. And this eikosogram is capturing kind of the joint probability of both of these categorical variates at once. Capturing that by having the marginal of the explanatory variate or the conditioning variate I along the horizontal axis, and then the conditional probabilities along the vertical axis of the response. So if we swap those around and made I the response and here the conditioning variate, you'd have the same probability distribution, but it would be expressed differently, because now we have the marginal probabilities of the hair color on the x-axis of the horizontal axis, and the conditional probabilities of the eye color given the hair color. So we're looking at this width being the marginal probability of the hair color being black, and this height of this first bar here, the conditional probability that the eye color is brown, given that the hair color is black. To make that easier to read off, if you actually want to read off the exact proportions themselves, we can just add axes. X-axis equals true, y-axis equals true, and now along the top horizontal bar, you can see that we've got a unit unit square here, and so we've got 0 to 1 on this axis, 0 to 1 on that axis, and now the probability that the hair color is black, the marginal probability is 0 to 0.18, so that's 0.18. The marginal probability that the hair color is brown is going to be 0.18 to 0.67, so the difference is that marginal probability. Similarly, the heights here, 0.06 is the conditional probability that the eye color is brown given the hair color was blonde, and 0.37 is the conditional probability that the eye color is brown given the hair color was red, and so on. Every one of these is identifying the height of one of these horizontal lines. And so the height of any one particular block is going to be found by calculating the difference between the two heights. So the eikosogram is actually showing the joint probability of these two categorical variates by specifying one as a response and the other as the conditioning variate. So one has its marginal distribution and the other has the conditional distribution display. We'll talk more about eikosograms in the next lecture.